We'll end off with the ear, and that's pretty much what we're going to focus on for special senses. So just the eye and the ear for lecture. The nice thing is that we've already looked at this in lab, so a lot of it is just kind of going over structures again, but adding a little bit of conceptual items to the list. So as far as the eye goes, the palpebra, okay, is a formal name for your eyelids. Okay, so you have an upper and a lower palpebra. The palpebral fissure is basically the space in between your upper and lower eyelid. So it's the space in which you see your eyeball. Okay. Canthi are basically the angles at either end of your eye socket. So you have a medial canthus and a lateral canthus. I'll show you the levator palpebrae superioris, which is on the next slide. That's basically just musculature on the top of the eyelid that allows you to physically open your eye. Okay. The conjunctiva is basically a mucous membrane that covers the whole anterior surface of your eye. And again, mucous membranes are designed to protect you against anything from the outside environment that wants to come in contact with your eyeball, of which there's all kinds of particulate matter that touches your eyeball all the time. You just don't think about it. And because it's a mucous membrane, it makes these little sacs on either above and below in the eye socket. Let's just go to the next slide because it's better for me to show you. Okay, so the levator palpebrae superioris is on the very top. So again, when that muscle contracts, it allows you to open your eyelid voluntarily. So that's going to be skeletal muscle. Okay. The conjunctiva, again, mucous membrane covering the anterior surface of the eye. It makes these little sacs. It makes a sac on the bottom, and it makes a sac on the top. Okay, so that's pretty much what I mean by sacs. And this mucous membrane can be irritated, and it can get inflamed. Okay. Does anyone happen to know the condition when your conjunctiva gets inflamed?
Conjunctivitis.
Conjunctivitis. Mm-hmm. Yeah, has anyone had conjunctivitis? Oh, I've had conjunctivitis. It's scary. So another story at my expense. When I was in college, did I tell you guys a story? The most anterior portion of the eye we already know is the the cornea. Okay, so the conjunctiva lays on top of that as a mucous membrane. That's where you put your contact lenses on top of your corneas. Okay. The lacrimal apparatus, basically what I want you to know is what's listed here in the order that's listed. So lacrimal glands, okay, are these exocrine or endocrine?
Exocrine.
Exocrine, because they use a system of ducts and they secrete their product locally, right? So lacrimal glands, okay, are going to produce the tears. Those tears are going to enter your eyeball from a lateral position, okay? So it's going to enter laterally. Tears are going to flow medially, okay? So you're going from the lacrimal caruncle to the lacrimal punctum. Now a punctum is a hole, and you have one on the bottom and on the top of your medial canthus, okay? So your medial and lateral canthus, medially, you can see in the mirror, you have two holes, okay? And those holes is where tears also drain, okay? The punctum then connects to what's called a canaliculus on the top and the bottom, okay? And then that canaliculus enters and empties into the nasolacrimal duct, okay? So that's why when you have a really good cry, like when you watch The Notebook or something, what happens to your nose? It gets all stuffed up, right? Because you're overloading the system. Okay? So, knowing what you know now about the lacrimal gland location and where tears are delivered into the eye socket, when you put an eye drop in your eye, should you put an eye drop in the area of the medial canthus or the lateral canthus? The lateral canthus, right? Because it gives the drop time to flow throughout the socket. Most people put it on the medial canthus. Okay, if you put an eye drop on the medial canthus, it's fine. You'll get it through the socket, but you're gonna put a majority through the punctum into the caruncle. Okay, so there's a little fun fact now that you know. Okay, so that's it for the lacrimal apparatus. Okay, extrinsic eye muscles we cover in lab, so you can just skip these 2 slides for our purposes here. Okay, as far as the eyeball itself is concerned, we know that there are layers that are involved with the eyeball as an organ. We have an outer fibrous layer, we have an intermediate vascular layer, and an inner sensory layer, which we call the retina. What do we call the vascular layer? The choroid. Right. And so we've got 2 different kinds of fluids in the eyeball, aqueous and vitreous, and they live in 2 different cavities, right? Aqueous for anterior, vitreous for posterior. Okay. The lens we've identified too, and we know the lens can change shape based on whether or not you need to see things close to you or further away. Okay, the lens tends to bulge when you need to look and focus on objects that are closer to you, and then it stretches out when you need to look at things that are further away from you. Okay, talk about that again. Let's talk about the different layers first. Okay, so the cornea and the sclera, which is the most superficial layer, are continuous with each other. Okay, and that makes up the fibrous tuning. Okay, fibrous connective tissue. Now on top of that, you've got your mucous membrane. Okay, to protect yourself against the elements from the outside. Okay, so we know that the sclera is the white of the eye. It's actually an extension of dura mater, of the brain, interestingly enough. So you are seeing brain matter when you look at the white of your eye. Okay, but because it's fibrous connective tissue, it gives an anchor for the musculature, the obliques and the rectus muscles that want to attach to it. Okay. So the cornea is the first structure where light is going to enter the eyeball, okay? And where the sclera here, and the cornea connect is called the limbus, okay? Now this picture is showing you really nicely, okay, the ciliary body, right? And the ciliary body has these little strings that are attached to it. What were those called?
Suspensory ligaments.
Suspensory ligaments, right? And that keeps the lens in place, all right? So the ciliary body also, and it's stated on a future slide here, I'm just getting ahead for a moment. The ciliary body also makes aqueous humor. So it also makes the aqueous humor that these arrows are depicting are flowing, okay, from the posterior chamber to the anterior chamber of the anterior cavity. Okay? So the posterior chamber of the eye is bounded by the lens and the iris, and the anterior chamber is bounded by the iris and the cornea. Okay? So aqueous humor is made by the ciliary body. It goes from posterior chamber into anterior chamber through what opening?
Pupil.
Through the pupil. And you're constantly making aqueous humor, right? Because it's a derivative of what?
Blood.
Of your bloodstream. Okay, so there has to be a way for you to drain it, right? So the eyes do not use the lymphatic system for drainage. The eyes have their own way of draining fluid. And what they use, but this structure you don't need to memorize, okay? But we know what a sinus is, okay? A sinus is a structure that's going to carry some sort of fluid, whether it's blood or in this case, aqueous humor, okay? You have on either side of the eyeball what's called a sclerovenous sinus, okay? Just like you have a coronary sinus in the heart, just like you have a superior sagittal sinus in the brain, right? Sinuses are going to drain something, and the sinus drains your eyeball. It drains aqueous humor and brings it back into circulation, into your bloodstream, right? Because if you didn't have a way of draining aqueous humor that's constantly being made from your bloodstream, your eyeball would do what? It would explode, right? So there has to be a way to maintain equilibrium of volume. Okay. Okay. So another condition in the eye, when pressure develops in the eye, if, for example, as you get older, your drainage doesn't work as well, and you're accumulating excessive aqueous humor in the eyeball, that's when they do that dreaded puff of air test that everybody hates, right, when you go to the eye doctor. But they're checking for the pressure within the eyeball to make sure it's within a normal value, okay? Because if it's not, then you can get increased pressure within the eyeball that can lead to variations in vision and ultimately blindness if it's not treated. Does anyone know what that condition is called? Increased pressure in the eye? Starts with a G.
Glaucoma.
Glaucoma. Mhm. Yeah, my dad had glaucoma, so we did eye drops for him twice a day. And he also had a laser procedure done, interestingly enough, because of course I ask all kinds of questions when I go to the doctor's appointments. And the laser treatment was designed to increase the size of the sinuses to allow for more drainage. So he basically went in, had a laser procedure done on the eyes that were diagnosed with glaucoma, and it was kind of a preventative procedure. Non-invasive, he didn't say he was in any pain, just a little bit light sensitive, but other than that, it was kind of a cool procedure. Okay. Okay, so going from the sclera, we meet the choroid, and the choroid is very vascular. Okay, so that's where you have all of your blood vessels, and it makes that layer very dark. Okay, so like we saw on the sheep or the cow eyeball, and that darkness prevents light scatter. So it helps collect light and hold on to it. It prevents light scatter. Okay. Okay, so we identified the ciliary body. Okay. And we know where the ora serrata is. It's this nice boundary here on the outside. So we identified the ora serrata on the models. Okay, we identified the suspensory ligaments. But now we know that the ciliary body makes aqueous humor. It's also smooth muscle. And it's smooth muscle that can contract and relax and allow the lens to either bulge or stretch out. Okay, so the ciliary body has control over the lens. Accommodation is just that, where the ciliary body can accommodate, where the lens can accommodate to whatever conditions are happening at the moment. Okay? Okay, the iris, we know is the colored part of the eye. So the iris also, though, is smooth muscle. And it has 2 layers of muscle that are associated with it. In the middle of the iris, we know is the pupil. And when you go to the eye doctor, they usually do the pupillary light reflex, where they shine a bright light into one eye, kind of covering the other eye. But interestingly enough, when you do the light reflex on one eye, the other eye will respond as well. And we'll talk about why in a minute. Okay, all right. So, the iris has 2 distinct smooth muscle layers. There's an inner sphincter layer, okay, that surrounds the pupil directly, and there's an outer dilator layer. Dilator, I think of like a sunburst or rays of the sun, okay? Sphincter is gonna be circular, right? Like your orbicularis oris or orbicularis oculi, circular musculature. So, in parasympathetic situations, when you're resting and digesting, like right now, you want to focus on things that are closer to you. So the sphincter muscle is going to contract, the dilator muscle is going to relax, it's going to make your pupils smaller so that you can focus on things that are closer to you. When the sympathetic nervous system is in control, fight or flight, the sphincter muscles relax and the dilator muscles contract, and that causes your pupil to get large, so you can see things further away from you. Okay. And again, I think I told you guys about my cat and my dad, how I used to sneak up on my cat and scare the bejesus out of him. And I would see his eyeballs, and man, his pupils were dilated. I'll never forget that. Like, that is a super sympathetic response in animals, is pupil dilation when they're scared. Okay. All right, so deepest layer is the retina. Okay, we saw that on the cow eye. It's almost like tissue paper. It's a neural layer, it's very thin, okay? And it basically has, well, it's got two layers that we need to address. The deepest layer is called the pigmented layer. Okay, so that's the deepest layer. Light is coming in from left to right here in the picture, so light is going this way. Okay, so the deepest layer of the retina is called the pigmented layer. And it's pigmented because, again, it's going to trap light. It's going to prevent light scatter. Okay? Okay. So everything else is considered the neural layer. Okay? So once light hits the pigmented layer, that pretty much ends the pathway of light. Okay? And we'll go over the two pathways tomorrow. We'll draw them, we'll write them out. We won't draw them physically, we'll write them out. Okay, so the end of the light pathway is signified by the pigmented layer. So once the pigmented layer traps and grabs those light wavelengths, we begin the electrical pathway, because that's going to stimulate these receptors, okay, called rods and cones. So rods and cones are responsible for different types of information, okay? And these are the only place where you're gonna find photoreceptors in the body. So cones are responsible for color, so C for color, and highlight situations. Okay, rods are more for low light situations, okay? So you have more rods than cones. So those photoreceptors get excited. When light waves hit the pigmented layer, boom, they immediately cause action potentials, electrical impulses in the rods and cones. And the rods and cones then connect to special sensory neurons that you only find in the eyeball. And structurally, what kind of sensory neuron are they? Bipolar sensory neurons. Yeah, that's what we're talking about. So that excites the bipolar neurons, which then stimulate and excite the ganglion cells. And that's the same rules still apply, because ganglia are collections of cell bodies, and we're talking in the eyeball here. So this is ganglion cells. So extensions of those ganglion cells are ultimately going to form axons, okay? And a collection of axons, okay, exiting the eyeball is going to form a nerve. What nerve are we talking about?
Optic nerve.
Optic nerve CN2. Yeah. So that nerve is going to send those those electrical impulses to the visual cortex of your brain, which is where? In the occipital lobe. For conscious processing of what it is you're seeing. Okay, so here again, the ganglion axons, okay, ganglion cell axons combine to form the optic nerve, and the optic nerve exits the eye at a region that we identified on the cow eye called what? The optic disc. So that's your natural blind spot, okay? You cannot process any information at the optic disc, okay? Because what's missing from the optic disc?
Rods and cones.
Rods and cones. Yeah, there's no receptors there to receive any information. So that's your natural blind spot. Okay. Okay, good. Okay. Ora serrata we've already covered. We know it's basically the end outer boundary of the ciliary body. The macula lutea is a region in the eyeball, which is where you guys were looking for what I mentioned, the fovea centralis, which is the area of most acute vision, okay? So the fovea centralis is found in a region of the eye called the macula lutea, okay? So people, as they get older, may experience macular degeneration, and that can affect the acuity of their vision. Okay. That's pretty much it from here. Okay, so nothing really new here, right? We've already looked at these structures in lab, which is nice. So we know that the posterior cavity has that jelly-like vitreous humor, and the vitreous humor is designed to keep the retina against the posterior wall of the eyeball, right? And vitreous humor, what you're born with is what you have for your lifetime. Okay. Anterior cavity is different. Within the two chambers, we have aqueous humor, which is constantly being produced as a derivative of your bloodstream. And it's going to be drained by those venous sinuses that I talked about. The lens, again, a disc. You guys were able to appreciate the shape in the cow eye dissection, okay? But a fresh lens is very pliable. It's very moldable, okay? The cow eye was hard as heck, but that's because it's a preserved eye. So our lens, again, is able to be more movable, more pliable, more bendable, more bulgeable, to allow light to be focused in certain areas of the retina. Okay, so this is probably the most conceptual thing on the eye that we're learning in lecture, all right? So all I want you guys to appreciate from the next two slides, okay, is how we process visual information, okay? So we're looking at a superior view of the brain, okay? So left eye is your left eye, right eye is your right eye, okay? Your left visual field is signified by the color green. Your right visual field is signified by the color purple, okay? Visual field meaning, okay, sagittal line going straight down the middle of your face. Your right visual field is what your right eye can see. Your left visual field is what your left eye can see, okay? Now, keep in mind, if you, for example, block one of your eyes, okay, and you're only looking through one eye, okay, do you see some magical, like, sagittal wall that prevents you from seeing things on the other side? No, you don't. Because your eye processes information from both visual fields. Okay, so each eye processes information from both visual fields, which is what gives you biocular vision. Okay, there's overlap, which is good. Okay, so posterior aspect of each eyeball, okay, is the retina. The big thick U represents the retina, okay? And coming out of the retina, we have the optic nerve, okay? Just like the X that we were looking at in the sheep brain. The middle of the X is called what? The optic chiasm, and that leads into the bottom of the X, which is the optic tract. And a tract is a collection of in the CNS, right? Mm-hmm. A nerve is a collection of axons in the CNS. Okay, all right. So, the optic nerve of each eye, okay, laterally processes information from the opposite visual field, okay? So the left eye, laterally, fibers are processing the right visual field. The right eye, laterally, these axons are processing the left visual field, okay? But the medial fibers process the same side visual field, okay? So both eyes are processing information from both visual fields, okay? Now, what you need to be aware of is we have this optic chiasm, and a chiasm is where there's a crossing over of information, okay? The medial fibers okay, of each optic nerve at the optic chiasm cross over. Okay, they cross over. And because they cross over, information that's in the left optic tract is only coming from the right visual field. Information in the right optic tract is only coming from the left visual field. Okay? And those optic tracts are going to synapse in the thalamus, big surprise, main sensory relay station, and those fibers are then going to terminate in the occipital lobe. Okay? So, true or false? The left occipital lobe interprets information from the right visual field. True. Okay? The right occipital lobe processes information from the left visual field. True. Okay? That's the concept that I want you guys to take away. So it's because the medial fibers cross over in the chiasm that then allows for all the information from the right visual field being processed in the left occipital lobe, and all the information from the left visual field is processed in the right occipital lobe. Okay? But each optic nerve, each eyeball, processes information from both visual fields, which is why when you close one eye, you can still see a whole host, okay, around you. You don't have just this wall that separates left versus right. Okay, that's what leads to that biocular vision. And that's why when the eye doctor does a pupillary reflex on you, your other eye is going to respond to that. So you'll get a response in both eyes. Okay, this is just adding some dotted lines to kind of drive home the fact that the medial fibers cross over, the lateral fibers stay on the same side. Okay. Okay. Questions about that? All right. I find that just talking that out makes more sense. If you're able to discuss it to yourself and tell it to yourself properly, then you've got the concept down. Okay, so these are basically in a list from top to bottom, giving you the pathway of vision. Okay, not light, but the electrical pathway for vision. Starting at the photoreceptor layer, ending at the occipital lobe. Okay? So in order, you should be able to know this. All the structures that get excited and stimulate the next person in line. Okay. All right, and we'll write that out tomorrow. I just wanted you guys to see ora serrata here. It's just kind of a nice boundary here on a cadaver specimen. Okay, questions about the eye. So not too bad. Most of the structures you already know. You know the cavities, you know the chambers, aqueous humor, vitreous humor, all that good stuff. Okay. All right, let's talk about the ear then. So same thing applies, all right? We've already learned a lot of the terms. So for the ear, we're gonna have an external, a middle, and an internal ear, okay? Internal ear mainly is gonna be involved in hearing. External and middle are going to be more involved in equilibrium. We'll talk more about that. So external and middle ear participate in hearing only. Internal ear functions for both hearing and I just said that the opposite way, I'm sorry. I said that the opposite way. External and middle ear participate in hearing only, and internal ear functions for both hearing and equilibrium. Sorry about that. In the internal ear, we're talking about the cochlea specifically. Okay, so external ear, the auricle is physically what you can see, okay? The helix is the rim. Typically you get piercings on the helix, also on the lobule. The external auditory meatus is the opening, and that leads into the external auditory canal. Nothing new there. You know, it's all just anatomy review. Okay, tympanic membrane, we know where that's located. That separates the external ear cavity from the middle ear cavity. So that's a boundary, okay? And it's designed to catch airwaves and transmit them to the auditory ossicles, which we already identified in lab. What's the first one? Malleus, and that hits the incus, and then finally the stapes. That looks like a horse stirrup. Okay, good. Okay, so the ossicles then are going to take those sound waves and transmit them from the middle ear cavity to the inner ear cavity through the oval window. Okay, the oval window is what the stapes is living on top of. Okay. Now, we don't hear all wavelengths of sound, all right? We as humans are sensitive to certain wavelengths. Other animal species are sensitive to other wavelengths, okay? So we have the round window, and the round window is designed to take wavelengths coming out of the cochlea that we're not processing and just dissipate them. Okay, give them a place to exit back into the middle ear cavity. Okay. So like both my dogs, I mean, if there is a squirrel or a possum or a bunny or something on the wall in the backyard, they are in the backyard before I even know what's going on, okay? Because they have a totally different wavelength of sensitivity to what they hear. And I just see them scurrying out, I'm like, you know, I get the after effect, like, okay, I missed that one. Okay. All right, so for the middle ear, we have the auditory tube, or the eustachian tube. Okay, the eustachian tube is designed to equalize air pressure on both sides of the tympanic membrane, because the external ear and the middle ear cavity are air only. Okay, the inner ear cavity is where we're switching to fluid environment. Okay. So we have a couple muscles that contract to protect us against sounds. Two of them. One's called the stapedius muscle, which is kind of nice because it attaches directly to the stapes. And then the other one is called the tensor tympani muscle, and that attaches to the malleus behind it and a little bit onto the tympanic membrane itself. And what those muscles are designed to do is when you're hearing really high frequencies or something potentially dangerous to the ear, they contract and they minimize the oscillations of your ossicles to protect you. And my husband and I, since we're getting older, we've started now using earbuds when we go to concerts, or if we go to a bar and listen to live music. I put protective earbuds in my ears to protect my ears against the loudness and the volume. And be careful with your earbuds too, because you have a tendency to want to blast, you know, music or whatnot. Be careful. You know, I'm noticing over time, my husband, he can't hear high frequency sounds. So there could be things beeping in the house, and I'm like, what's beeping? And he's just like, I don't know, what is it? You hear something? Like, yeah, I hear something. There's something beeping. It's upstairs. What's going on? You know, it's one of the fire alarms or something alerting us that there's a low battery. He can't hear that. I'm just like, okay, that's concerning. Like, we need to make sure that we're protecting our ears. So, keep that in mind for you guys. You're young now, but as age starts to hit you, be careful. Okay. All right, so the inner ear, we're going to talk a little bit more detail about. Again, this is going to be a fluid environment, okay? We've talked about a lot of the anatomical structures before, but now we need to know what's their purpose. Why are they there? What's the function? Okay? So in the cochlea, all right, you have a bony labyrinth. So you have bony structures, okay? You have bony structures, like your semicircular canal, your vestibule, your utricle, your saccule, okay, and your cochlea, okay? So, that's superficial, you have your bony labyrinth. Deep to those structures, and light blue in this picture, you have soft membranous structures, okay? So within your semicircular canals, you have semicircular ducts. Within the vestibule, you have soft membranous structures called the utricle and the saccule. The vestibule is the bony structure that surrounds it. Cochlea, deep to that, we have the cochlear duct, which we also call the scala, what? Scala media, right? Same thing, so it's called the scala media. Okay. All right. Okay. Scala media. Okay. All right, so in between the bony labyrinth and the membranous structures is going to be perilymph. Okay. So deep to the semicircular canals, but superficial to the semicircular ducts, there's perilymph that's flowing. Okay. In the soft membranous structures, we have endolymph. Okay, so in the cochlear duct, we have endolymph. In the scala vestibuli and scala tympani, we have perilymph. Okay. I'll show you another picture of this in a minute. Okay. So the best way to describe it, for those of you that are visual, like if you're not getting this, okay, I'm just drawing a couple fish in a fish tank, okay? So, the tank is the bony structure, okay? In the tank, you have water. That's the perilymph, okay? Then you have these soft membranous structures floating around, okay? Those are the fish. And in the fish, you have endolymph. Okay, so that's what I mean by the different layers, okay, and the fluid that lives within them. So if that helps you with the layers of fluid versus structure, okay? Okay, semicircular canals, semicircular ducts, responsible for rotational equilibrium, so your ability to look right and left. Those are semicircular canals' jobs, to interpret that type of information. Okay? The utricle and the saccule are going to be responsible for you recognizing when you're static, when you're not moving, and also linear acceleration of your head, so whether you're looking down at the floor or you're looking up at the ceiling. Okay, that's the utricle and the saccule's job. And the cochlea has nothing to do with equilibrium. Cochlea is all about hearing. Okay. 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 So let's talk a little bit about each part. Okay. So the vestibule is the bony structure. Deep to that is the soft membranous structures called the utricle and the saccule, okay? So in this picture, okay, you've got the big vestibule, okay, that sits in between the semicircular canals and the cochlea, and the big vestibule. Inside of that is soft membranous structures called the utricle and the saccule, okay? And each one of those, the utricle and the saccule, has special epithelium called the macula. And this little purple glob in each one is the macula, okay? So the macula is epithelial tissue, okay, specialized epithelial tissue that has hair cells that come out from its apical surface, okay, hair cells. And those hair cells are going to embed themselves in some sort of gelatinous membrane. And this is going to be a repeating concept, okay? So, the membrane here is called the otolithic membrane, and it's made of calcium crystals, okay? But the epithelial tissue that houses the hair cells on the opposite ends have nerve endings. So the hair cells can get excited when they get stimulated. Okay? So how do they get stimulated? Okay, well the utricle and the saccule is responsible for static equilibrium and linear acceleration. So looking down at the floor, which is the example, or looking up at the ceiling. When you look down at the floor, okay, that's going to move fluid. It's going to move the endolymph. Okay? The endolymph is going to pull on the membrane. It's going to pull on the autolytic membrane. The autolytic membrane is going to pull on those hair cells. It's going to excite those hair cells, which is going to send electrical impulses down the vestibulocochlear division of cranial nerve. What number? 8. Yep. Going to your cerebral cortex so that you have the conscious perception of the fact that you're looking down. Okay. Now, don't worry about horizontal versus vertical. That doesn't, the difference, don't worry about it. I just want you to know that there is the macula, which is a specialized epithelium that has hair cells, that have nerve endings, with a membrane on top. The membrane moves, it pulls the hair cells, it excites the nerve, sends electrical impulses to the cerebral cortex for conscious level processing of what it is you're doing. Okay? So people that have vertigo, okay? If you know anyone that's had vertigo, this is where the membranes get ruffled up. Those calcium crystals get messed up, usually from some sort of a trauma. My roommate in PT school, actually, she went out bodyboarding, boogie boarding, and she caught a really heavy wave and it basically knocked her head into the sand. Thank God she didn't break her neck. But it gave her temporary vertigo because it was such a forceful impact that it totally messed up her inner ear. And it's awful for these people because you feel like you're on a roller coaster when you're not. Your whole interpretation of your orientation and where you are in space is totally messed up. So I mean, you could be sitting in a chair, but you feel like you're on a roller coaster and you just want to throw up. Like it's just horrendous. It's horrible. It's awful. So there's actually a specialized division of physical therapy that is purely vestibular that helps these people get back to normal. And it's learning a system of maneuvers, getting the patients in certain positions, including rotational movements and linear movements in an effort to reposition the membrane and the calcium crystals back into a normal alignment to give these people relief. I can't imagine feeling that way. You can't function. You can't drive, you can't do anything. Okay, so semicircular ducts. Those are the soft membranous structures that are within the semicircular canal, right? And those are responsible for rotational movements, which is why we have 3 different orientations of the semicircular canals. Like one's in the sagittal, one's in transverse, one's kind of in an oblique angle, okay? Okay, so in each semicircular duct, we have specialized epithelia called the crista ampullaris, okay? The crista ampullaris has hair cells. Those hair cells have nerve endings, okay? And the gelatinous structure that the hair cells embed themselves in is called the cupula, okay? So you rotate your head to the left or to the right, endolymph is going to move the cupula, which is going to pull on the hair cells and fire off those nerve endings, sending impulses down the vestibulocochlear division of your cranial nerve to the cerebral cortex for conscious level processing of the fact that you're rotating your head to the right or to the left. Okay. Okay. Last bit here is the cochlea. Okay. So now we're switching from equilibrium to hearing only. Okay, this is hearing only. wow, these look a lot like that one model that you saw yesterday on the practicum. And we also looked at it under the microscope, right? Okay. So the cochlea is basically a tube that just kind of wraps around itself, which is why you see a bunch of these same structures. It's just a tube that wraps around yourself, so you're just kind of seeing the same thing over and over again. And the modiolus is just a name, you don't have to memorize that, of what they call that bony interpretation. Okay. All right. No surprise here, okay? You wear your vest on top, panties on the bottom, right? Both of these guys, perilymph. Scala media, what kind of fluid? Endolymph. Okay, so this is a soft membranous structure. These are in between the soft membranous structure and the bony structure. Okay, so that's going to be perilymph. We know vestibular membrane. We know basilar membrane. Okay? So nothing new there. But now we've got to look at, okay, how does the cochlea take information about what it is you're hearing? Okay? Well, it has a specialized structure. We didn't identify the structure in lab, but we identified some of the parts. Okay? The organ of Corti. The organ of Corti is specialized epithelia that has hair cells that are coming out of it. Those hair cells have nerve endings. Okay? And those hair cells embed themselves in a special membrane that you guys had to identify in lab called what? The tectorial membrane. Okay. So, sound waves come in, vibrate the endolymph. Okay? It's going to cause the tectorial membrane to move. Tectorial membrane is going to pull on the hair cells, firing off nerve impulses that are going to go down the vestibulocochlear nerve, specifically the cochlear division, because it's just about hearing. And that's ultimately going to terminate where in the brain? Where is your hearing? Auditory cortex.
Temporal.
In the temporal lobe. So that you get consciousness of what it is you're hearing. Okay? Okay. So, the hearing pathway, to put it, just kind of put it all together. Okay? So your ear, external ear, is designed to pick up airwaves. It's designed to pick up airwaves and put it into the canal. Okay? So airwaves enter the canal and they hit the tympanic membrane. What's another word for your tympanic membrane? Layman's terms? Your eardrum, right? So don't put a Q-tip too far in there, right? You don't want to damage your eardrum. And you don't want to get rid of too much wax, right? Remember, wax is a good thing. Okay, so that's going to vibrate the tympanic membrane, which is going to vibrate 1, 2, 3 ossicles. And the stapes is attached to the oval window. So here we're going from air only, now we're going into fluid. Okay, so the oval window opens up into the scala vestibuli. And it's going to vibrate the perilymph. Okay? And within whatever wavelengths we can hear and we're sensitive to, those wavelengths are going to enter the cochlear duct. Okay? And the cochlear duct, we're moving from perilymph to what kind of fluid? To endolymph. So the endolymph is going to start to vibrate. It's going to pull on the tectorial membrane. It's going to fire off the hair cells. It's going to send electrical impulses down the cochlear division of the vestibulocochlear nerve, terminating in your temporal lobe, so that you have conscious level processing of what it is you're hearing. Now, we don't hear all wavelengths, okay? So some of those wavelengths just keep on going, and they don't enter the cochlear duct. They just do a drive-by. And those wavelengths exit out the round window and dissipate. Okay? And that's the pathway of sound. crazy to think about. I mean, you can think about how fast this pathway occurs and how often. Okay. So again, you don't have to memorize the pathway. This is just pulling it all together. Okay, pulling it all together. So we're in scala media. You've got tectorial membrane with the hair cells. Okay, nerve endings. Okay, these look like sensory bipolar cells. Okay, you only find these in the eye and the ear. They're going to come up, bringing in sensory information. They're going to synapse in deep clusters of gray matter in the CNS, where we find synapses in cell bodies and things. This is communication, we already know this. It's going to send information up because it's a sensory pathway. It's going to synapse in the thalamus, which is the main sensory relay station, and ultimately it's going to wind up in your auditory cortex in the temporal lobe. And only when it reaches there do you have conscious perception of what it is you're hearing. All based on vibration of sound. Kind of crazy. Okay. All right, that's all I got. Questions?